All right, in our previous chapter, we talked about cell structure and function. In this chapter, we're going to focus specifically on the membrane because it plays such a vital role to maintaining efficiency across the cell. So we're going to talk about the plasma membrane structure and function, then we'll talk about passive transport across the membrane, then followed by active transport across the membrane, and then we'll talk about some modifications to cell surfaces. Starting off, we're going to just talk about the plasma membrane as a whole. As you may have noticed in our previous chapter, when we talked about prokaryotic and eukaryotic cells, we're going to find that the plasma membrane is found in both. Not only is it found in both, it's found in all. All cells have a plasma membrane. Its primary goal is going to be to separate the internal cytoplasm from the external environment of the cell, and the membrane surrounding the various organelles and components of the cell make it possible for incompatible chemical reactions to occur simultaneously. This division of labor allows cells to operate more efficiently even if they are larger eukaryotic cells. So when we're talking about the cell membrane, you're often going to hear the term fluid mosaic model. This depiction of the plasma membrane is called that fluid mosaic model because of the various proteins, lipids, glycoproteins, and carbohydrates that are going to be embedded throughout the membrane. That gives it kind of that very unique mosaic landscape. However, the mobility and flexibility of the phospholipids gives so much freedom and flow to movement that it has the appearance of being almost fluid in nature. You can kind of think of it, a lot of times these peripheral proteins are going to be they're going to be embedded in the plasma membrane, and they're going to be attached to the cytoskeleton beneath it. So you can kind of think of this plasma membrane as being an ocean moving around buoys, or ships that are anchored. Those ships or buoys are going to stay anchored, tethered to one particular position, but the water can move and flow around it. So we're going to have very, very fluid movement, not only in the phospholipids, but any protein that isn't anchored to the cytoskeleton on the other side is also going to be able to move and flow in any direction. So interestingly enough, at body temperature, the plasma membrane has the consistency of olive oil. So it's going to be kind of a very slippery fluid substance. The phospholipid bilayer has several unique characteristics that we've already covered, but are worth mentioning again. Like we said before, it does have that phospholipid bilayer where that external surface is going to be lined with the hydrophilic polar heads. The cytoplasmic surface is also going to be lined with the hydrophobic polar heads. The nonpolar hydrophobic fatty acid tails are going to be sandwiched in between. And the double-bonded fatty acid tails prevent close packing. It helps contribute to the fluidity of the membrane. And the greater concentration of those unsaturated fatty acid residues, the more fluid in nature the structure of the membrane is going to be. One last thing I want to point out is that these monolayers can actually move past each other. So you can kind of think of it as a sandwich, two pieces of bread with a very, very thick, slippery layer of jelly in between. Those two sandwiches can actually slide past each other and move really in any direction. So I'm going to talk just a little bit greater in depth about the three components of the plasma membrane. Like I said, cell membranes are going to be very similar in the types of molecules that they contain, which makes them interchangeable and allows them to fuse together very, very easily. And we discussed this a little bit in the last chapter when we described how vesicles are formed and transferred through the various endomembrane organelles. Just for example, proteins are synthesized by the ribosomes in the rough ER. They travel through the ER until the membrane pinches off, forming a small vesicle around that protein. That vesicle from the ER can then travel to the Golgi apparatus, fuse with the Golgi apparatus, travel through the Golgi apparatus, and then pinch off in a vesicle on the other side where it can be transported to the cell membrane. Once we reach that cell membrane, the vesicle will fuse with the cell membrane and release its contents to the world outside. So while it is useful that the cell membranes can merge and are somewhat interchangeable, there are also instances when it's important to differentiate those types of organelles. So in that case, we're going to look for protein molecules that are going to help us differentiate the different types of membrane. So there are different types of proteins that can be either integrated in the membrane either partially or wholly. Peripheral membranes are going to be proteins that only occur on the cytoplasmic side of the membrane. They're only going to be found on the inside of the plasma membrane. Integral proteins are going to be embedded either partially or wholly through both layers of the plasma membrane. And those integral proteins are going to be held in place by the hydrophobic, hydrophilic regions on that protein. Or they're going to be attached to the protein fibers of the cytoskeleton. And not all proteins are going to be held in place by the cytoskeleton. Some are able to move sideways through the through the plasma membrane. So some will be docked, some will be anchored to the cytoskeleton, but some will move freely. The last component of the plasma membrane is going to be cholesterol. Cholesterol is actually a really important part of the plasma membrane. It's going to be a lipid steroid that's found in the plasma membrane that contributes to the fluidity and rigidity of the membrane across various temperatures. In other words, it prevents the membrane from becoming too fluid at high temperatures, so it helps the plasma membrane maintain some of its sturdiness and its structure. But at cooler temperatures, it also prevents the membrane from freezing. It prevents the really close packing and contact between those phospholipid tails. So it sort of keeps the plasma membrane nice and open. So here is our cholesterol, which is going to prevent these phospholipid or these hydrophobic tails from packing in too close to each other. Now there are a couple of other key components that are found in that plasma membrane. There are going to be certain molecules like glycoproteins and glycolipids that are found in the plasma membrane and play a very important role. For example, there will be carbohydrate chains found on the cell's exterior. So these glycoproteins and glycolipids are going to be found only on the outside of the cell, not on the cytoplasmic side of the cell. And the reason why they're found on the outside of the cell is because they contribute to the cell's fingerprint. The two G's play a very important role in cellular identification. These chains are going to vary in number in the sequences of sugar and in the branching structure of those carbohydrates. And every cell within an individual has its own fingerprint due to these individual markings. So because of this, glycoproteins and glycolipids play an important role in cellular identification. These are what are going to help identify the cell as, oh, this belongs to me, it's all one organism, this is in particular a liver cell, and we need to make sure that it's where it needs to be and performing the functions that it needs to do. These glycoproteins and glycolipids actually do play an important role in tissue transplantation and blood transfusions. This is why we talk about having a match when it comes to transplants or transfusions. Any cell that does not have the correct identifying fingerprint is going to be marked as a foreign invader and treated as such. So it's very important that we match those glycoproteins and those glycolipids. Glycoproteins are going to be proteins that have a carbohydrate attached to it. A glycolipid is going to be a lipid that has a carbohydrate attached to it. So these carbohydrate chains exist only in the outside membrane of the cell. Of course, it's kind of like wearing a name tag on the outside. You would never wear a name tag on the inside of your shirt because who needs to see that? Just you, and you know who you are. You do not need to see your own name. The name tag is for everybody else around you to see it, to know who you are and identify who you are. All right, so we've talked about sort of the different components that are found in the plasma membrane. The next thing we really want to get into is the plasma membrane proteins. The plasma membrane proteins are going to be found, like we said, either partially or wholly embedded in the plasma membrane, and each one of them is going to have its own particular function. For example, we're going to start off with channel proteins. Channel proteins are pretty much exactly what you would expect them to be. So although the protein's composition of the plasma membrane differs from one cell to the next depending on the type of cell and its current role, there are a few types of proteins that are likely to be present in every cell. So like I said, we're going to start off by talking about channel proteins. They're going to be involved in the transport of molecules across the membrane. They're going to form a channel for any molecule that is too large or too charged to get through the phospholipid layer by itself. Very small non-charged molecules like oxygen or carbon dioxide can diffuse and squeeze past these phospholipid bilayer to go in and out of that plasma membrane. Any molecule that's too large or too charged won't be able to make it and is going to have to travel through a channel protein. So a great example of this is that there is a protein channel that is responsible for the movement of hydrogen ions across the mitochondrial membrane, which is going to be absolutely necessary for the formation of ATP. And we're going to talk about that quite a bit when we get into cellular respiration. Another type of plasma membrane protein would be a carrier protein. Carrier proteins are another type of transport proteins. These proteins bind with the molecule they're transporting and then alter their shape to allow the protein to move across to the other side. These are going to assist in the passage of molecules through the membrane. So it's kind of like I don't know, when you enter a double set of doors. You walk in through the first set of doors, those close behind you, and then you go in through the second set of doors. That's what's going to happen with these carrier proteins. They're going to open up on one side of the plasma membrane, bind to their molecule that they're transporting, and then open up on the other side of the plasma membrane in order to pass them along. Next up, we have cell recognition proteins, things like glycoproteins. Glycoproteins, as I've already mentioned, they contribute to the overall fingerprint of the cell. They help the body recognize friendly versus foreign substances. Then there are going to be receptor proteins. These are a very important membrane protein that are going to be involved in cellular communication. Receptor proteins each have a unique shape that will only allow certain molecules to bind to them. When the correct molecule binds to the receptor protein, it's going to undergo a physiological change just like the carrier protein does. However, instead of releasing that molecule on the other side, this conformational change or this structural change is going to signal a response and change in the rest of the cell. So what happens is this receptor or this signaling molecule is going to bind to this receptor, and then this cell is going to send off a signal cascade that's going to change a cellular response in the rest of the cell. This is how a lot of hormonal signaling in the body occurs. A hormone molecule will bind to a receptor at the surface of the cell, which is going to signal a response inside of the cell. A great example of this would be the PIK3 hormone signaling. Here's a little bit of how that cell signaling works. Here we'll have a receptor, or we'll have a signaling molecule that binds to the receptor. Now this signaling molecule does not get transported across the membrane. We're not trying to get that signaling molecule inside. We are just ringing the doorbell. We are going to activate the receptor on this side, which is going to start off a transduction pathway. It's just going to be a series of relay proteins that end when the protein is activated. So we're just carrying the message along like a telegraph. Once we have gone down our transduction pathway, we're going to then activate a protein at the other side. This could be structural protein, which would change the shape or cause movement of the cell. It could be to activate an enzyme to either speed up or slow down metabolism. Or it might be a gene regulatory protein. Maybe we need to upregulate a particular gene or downregulate a particular type of gene to produce more or less proteins. I know I didn't spend a lot of time on this, but there are so many amazing videos out there about cell-to-cell signaling, about transduction pathways. So I intentionally kind of left this a little bit vague because I would love for you to do your own research and send me some tutorial videos on cell signaling and transduction pathways as you find them out there. These would be great extra credit opportunities if you're going to be sending me those extra credit videos. So please explore a little bit on the cell signaling pathway. Another type of plasma membrane protein that I want to mention would be enzymatic proteins. These proteins are going to be responsible for carrying out metabolic reactions directly. Just like any other enzyme, they just happen to be bound to the plasma membrane. And then finally, junction proteins. Junction proteins are going to be responsible for attaching adjacent proteins, they attach to adjacent proteins and they're going to attach cells to each other. We're going to see junction proteins play out a little bit at the end of this chapter. All right, and that is going to be where we're going to pause for now, and we'll pick up with talking about the permeability of the plasma membrane.